Okay, whew, had a little trouble getting started there. I had to change my password and wasn't finding the network, but I think we're good. We got going. So I hope you all had a good break. We got 6 more weeks to go, so seems doable. I know it's kind of a hard stretch at the end of the semester, but hang in there. Our next exam is a week from today. So right now, let's see, I'll get something out to you about that in writing, but I'm planning on that covering the viruses. That was chapter 13. Chapter 9, that was antimicrobial methods. And can you hear me okay? I know it blows loud back there. So, also chapter 10, antibiotics. We're going to finish that today. And I was planning on including the next chapter, chapter 14 on infection. I'll evaluate that after today and what I think we can get to on Wednesday, and we'll see how that goes. So, yeah, so that's looking forward what we're going to be getting into. We started antibiotics last time. We talked about different things that we have to keep in mind when we're developing antibiotics, and are they selectively toxic? How do we administer them? All that stuff. And we got through a number of antibiotics, went through all our beta lactams, our cell wall inhibitors, and we started the protein synthesis inhibitors. So remember, as we go through these drugs, again, we're going to finish those today. Think about number one thing you need to know about each one of the drugs is how they work. We call that the mechanism of action. So paying particular attention to what structure do they target on bacteria that we don't have, what makes them selectively toxic. Also think particularly about the uses, like if we have a particular thing that we use streptomycin for instance. Pay attention to that, and that's not something that I'm always super clear on, but pay attention to that when I bring it up. And also if there are disadvantages or big advantages to using the drug. So for instance, with streptomycin, we talked about the toxicity to the ears. So it targets cranial nerve number 8. So that's a huge problem, and not what keeps us, in addition to a lot of organisms being resistant to it, but that's mainly what keeps us from it being in wide use any further. Okay, so a lot of other things I might talk about, like for this one, I said, well, it's a natural product of a certain genus. Sometimes I've thrown some things in to be interesting. Sometimes I've thrown some things in just to remind you, hey, some antibiotics are natural products, some are semi-synthetic, some are fully synthetic. How they're delivered, mostly to get you to kind of remember, hey, how we use those terms. What does acid labile mean? Okay, so sometimes on exams, I like to ask questions. Oh, here's a drug that we use, and this, and this about it. So I might give you lots of clues to the one that I'm talking about. Okay, and I will post some practice questions here in the next few days once I figure out for sure. I've got sort of I've done a lot of work on the exam already, but we're getting there. Okay, so streptomycin was an example of an aminoglycoside. There are other protein synthesis inhibitors. The tetracyclines are another big group that target the small unit of the small subunit of the bacterial ribosome. Tetracycline, when you look at the structure here, 4 rings, tetracycline. Wow, that makes sense. Okay. And how does that work? This also targets the small subunit. Let me come back to this page in a minute. Your book's diagram of the mechanism of action there kind of makes it look like it binds to the messenger RNA during translation. No, it actually binds to that small ribosomal subunit, but it binds to the place where the new charged tRNAs come in to dock to make that new protein. So it actually blocks that docking site so that the growing peptide can't grow anymore. Okay, so it's a fairly, like most of the protein synthesis inhibitors, they're fairly broad spectrum, meaning they work on gram positives and gram negatives. They're really cheap to make. So that's one of the reasons that tetracyclines have been used so broadly in agriculture, just added to feed, not only to keep animals from getting sick, but we know that when we add antibiotics to livestock feed, they actually grow faster. So this is something we're trying to get away from because we think this this overuse of antibiotics contributes to the rise of antibiotic resistant infections. We'll talk more about that shortly. But they have been, especially tetracycline, used extensively in agriculture. They can be given orally by pill form. So they don't have to be injected. So they're, stable in stomach acid. The really jarring side effect that we see from tetracycline is that it can stain developing teeth. All right, so, not a nice picture there. So for that reason, we don't give any tetracycline drugs to kids or pregnant women. Also, and so there are tetracyclines that are in common use. Doxycycline is one that's pretty commonly prescribed for things like pneumonia, or if somebody is suspected to have Lyme disease, for instance. Also, a big use in acne medication. So you may be on an acne medication, and your doctor may have said, make sure when you go outside to use sunscreen. Okay, tetracyclines are known to have a little higher phototoxicity. That just means you're more likely to get sunburned, especially if you've got a topical cream on, or even if you're taking it by pill. In addition, tetracycline, any drug that you take, it has an expiration date on it, and don't take it after the expiration date. Usually you're advised to throw it out after the expiration date because the drug just degrades and it's not going to be as useful. It's not going to work as well. But tetracycline, actually, when it degrades a little bit, it becomes more toxic. Okay, so in this case, you'd always want to throw it out when it's past the date. So uses, I mentioned a few there. They've been widely used in agriculture, and a few other specific things like prostate infections or intracellular parasites. Okay. Let's see. One more protein synthesis inhibitor or class of drugs that are protein synthesis inhibitors, the macrolides. And the mechanism of action here is a little bit different. It actually binds to the large subunit of a bacteria instead of the small one. Okay, so when it binds there, it just, the messenger RNA can't move through the ribosome as easily, and so it's typically going to stop protein synthesis. In structure here, like I said, don't memorize structures. I like to point out things that are just sort of, hey, this makes sense. This whole ring here, that's called a macrolide ring. I wouldn't necessarily expect you to know that, but that's why we call them the macrolides. There's really only a few drugs in this group. One very, very common though, azithromycin. About 10 years ago, that was the most common drug prescribed for outpatient ailments. So a lot of you have probably taken a Z-Pak, or it's also called zithromycin at some point, Zithromax. It's a really kind of nice, easy drug to take because it comes in a packet of 5 pills. And you take one pill every day for 5 days, and usually that's enough to kind of get rid of sort of some of our common ailments. So things like strep throat or ear infections, especially commonly given to patients if they have a penicillin allergy. So they can't take amoxicillin for some of those general things. It's also used in conjunction, usually with a tetracycline for gonorrhea. Early on in the pandemic, azithromycin was kind of put forward as when we were searching for drugs that we already knew were safe to see if there was one that might be effective against COVID. For a time, we thought maybe azithromycin might be helpful against COVID. And that really, when I heard that, I thought, well, that's crazy. This is an antibiotic. And, obviously COVID is viral. Why would that make any difference? Azithromycin actually has some anti-inflammatory ability, as well as it kind of has been shown to increase some aspects of our immune response. So early on, we were hoping that that might be helpful, but I believe that they found that not really to make any difference at all. So in case you've heard of that before and wondered that. But it's fairly broad spectrum against bacteria, some gram positive, some gram negative. Like I said, we can take it by pill, and you only have to take it once a day. It stays, it reaches an effective concentration pretty quickly, and it doesn't degrade very fast. So we only have to take it once. So that's kind of nice. There are a few allergies to it, not as many people as have a penicillin allergy. Some GI issues like, a little nausea. Not nearly as bad as sort of the earlier generation macrolide erythromycin. That was a lot worse, a lot more stomach cramping when people took that. So this is a little bit better. Okay. Don't be afraid to jump in if you have questions or comments. We have a DNA inhibitor and an RNA inhibitor. So rifampin, or sometimes it's called rifampicin, is an RNA inhibitor because it blocks that RNA polymerase. And I think it binds to the enzyme itself. Remember, bacterial RNA polymerase is a little bit different. In eukaryotes, we have a different polymerase, RNA polymerase, to make different kinds of RNA. It's a semi-synthetic. It's really easily absorbed, so we can take it either by pill form or it can be injected. It does have sort of the disconcerting side effect because the pill itself is an orange color. It will change our bodily fluids orange. Okay, so here's a picture of somebody's catheter bag that was on rifampin. So you can see how orange it's changed that person's urine. It's supposed to be totally harmless. It's just, like I said, sort of disconcerting. And like with a lot of antibiotics, we get, some nausea, some vomiting, some loss of appetite as a side effect there. Although rifampin can be used for a lot of different infections, we primarily use this. It's one of the first drugs that we use to treat tuberculosis and leprosy. And when it's used to treat TB, we use it in combination with other drugs. We talked about the other drug, isoniazid, which interferes with mycolic acid synthesis. So we usually would use isoniazid and rifampin together, as well as usually a few other drugs to treat tuberculosis. Okay, so it's one of the drugs that just kind of penetrates through that cell wall pretty well. Let's see, make sure I don't forget to say anything here. All right, and a couple of DNA inhibitors. So the quinolones, those are sort of the first generation drugs, and the fluoroquinolones are kind of the second generation drugs that target DNA gyrase. So remember, that enzyme is the one that's upstream of the replication fork. Let's see if I can draw that. Okay, so as those two DNA strands are being pulled apart, we get supercoiling up here upstream. And so bacteria need that DNA gyrase to cut each strand of DNA, kind of unwind it and seal it up again. So the quinolone and fluoroquinolone drugs bind to that DNA gyrase and keep it from sealing the DNA strands up again. So basically you get this fragmentation of DNA that it proves fatal to the cell. Bacterial DNA gyrases are different enough than ours so that it can be selectively toxic. So the quinolones, they're not in use much anymore, but an example of a quinolone is nalidixic acid. So maybe that sounds familiar to you if you had lab already this morning, but we use a media that has nalidixic acid in it that inhibits gram-negative growth. Okay, so this mostly works on gram-negative. Again, those were kind of the early generation drugs. We found that if we alter those quinolones just a little bit by adding a fluorine atom there, that it just makes the drug bind even better to the DNA gyrase, and it just works a little bit better. And what's the other thing I was going to say about that? Oh, right, it improves, it helps it penetrate into the bacterial cell a little bit better. So look, fluoroquinolones are more like kind of the second generation quinolones. And they are in very common use. Ciprofloxacin is probably the most commonly recognized fluoroquinolone. They all end in this suffix oxacin. So there are a number of fluoroquinolone drugs, often called Cipro for abbreviate that. So the fluoroquinolones are more broad spectrum, where again, the quinolones mostly just works on the gram negatives. They're absorbed pretty well and stable in acid, so that means we can give them in pill form. Although these fluoroquinolone drugs are not typically used as kind of the first choice. Like somebody's got an ear infection, you don't give them a fluoroquinolone. You wait and see if nothing else would work, then maybe you would use that. But generally not even for something as inconsequential. Not that the ear infection can be painful, but it's not life-threatening, basically. Because we see some really nasty side effects that, although they're rare, they're, like I said, particularly nasty. So we generally are not going to try a fluoroquinolone as our first option, only after our options are kind of run out. So for instance, anthrax, Cipro is often given if somebody's been exposed to anthrax and we want to keep them from starting to show symptoms. Some maybe a particularly tough urinary tract infection that wasn't responding to other drugs, or a particularly tough respiratory infection. About 5 years ago, the FDA put out a caution that we really shouldn't be using these drugs unless we absolutely have to. It said it's like using one epidemiologist said it's like trying to kill a fly with an automatic weapon. Okay, so we don't want to use these unless we really have to, because some of the side effects we see, this is a really bizarre one, tendon disruption, especially in older adults. Achilles tendon. So imagine taking an antibiotics and tearing your Achilles tendon as a result of it. That's kind of a bizarre thing. We don't really understand why that happens. It maybe the antibiotic impedes drug flow to joints or impedes collagen formation. We don't really understand why, but you know, that's a pretty serious side effect to keep you laid up for even longer. And in kids, we don't give fluoroquinolines to kids because there's some rare but neurological side effects. So obviously that should be avoided. Aortic rupture, right? Pretty serious side effects. So although they're rare, you know, we try to avoid that and only use these when nothing else is working. And I don't think I posted this, but every once in a while I come across a mnemonic that seems like, well, this may be helpful. So the queen stops the gyrating dancers. So gyrating, DNA gyrase, queen for quinolone, stops DNA gyrase. I don't know, may be helpful. Mechanism there. All right, two more mechanisms to explore a little bit. Cell membrane inhibitors. We don't have very many of these because, remember, we're looking for something that's selectively toxic, and our membranes are very, very similar to bacterial membranes. So the problem here is toxicity issues. So when we ingest a polymyxin antibiotic, that just is usually too much for our kidneys to handle. We get too much kidney toxicity. So when they are used, they're only used topically. Okay, so for skin infections, unless somebody's, you know, life is at stake and nothing else is working, then they may give it by IV. But again, typically a topically used drug. And so an example of a polymyxin colistin, right? So again, if you had lab already, this might look familiar, but we use a media with colistin and nalidixic acid in it, a couple of different antibiotics that are used to inhibit gram-negative growth in our Petri dish. Colistin, again, is a polymyxin antibiotic. And if you look at the structure here, you can kind of make out the ring and the tail. That might remind you of a soap or detergent. And we talked about how those work. They insert into membranes. This hydrophobic tail inserts into membranes. So kind of the same idea there. Particularly useful against gram negatives. Remember, they have that outer membrane, but that tail will insert all the way into its plasma membrane as well. The main use that we have for polymyxins is for skin infections. So it's that third drug in triple antibiotic cream. Skin infections. So remember the other two that we talked about, bacitracin, cell wall inhibitor, neomycin is an aminoglycoside, a protein synthesis inhibitor, and now it's actually polymyxin B. All right, so you've got three different mechanisms that are pretty useful against a lot of different skin infections. Okay. All right, so I know I've run through a lot of different drugs. We've got one more to talk about, and there is really one really good example of a drug that we have that can target a metabolic pathway that bacteria have that we don't. Okay, and those are our sulfa drugs. So if you look at the mechanism here, this is the pathway that bacteria use to synthesize folic acid. Okay, and folic acid is necessary not only for us, but it's necessary for bacteria, let's see, to make DNA. So needed to synthesize DNA. It's actually a coenzyme that's necessary for some of the enzymes to synthesize DNA. So if you look at that pathway a little bit, to simplify a little bit, the substrate here, para-aminobenzoic acid, PABA. Okay, that's one of the substrates. And there are several different enzymes that bacteria have that synthesize, that help the cell and end up with folic acid. So one of those enzymes here, don't memorize the name of that, but the sulfa drugs or sulfonamides inhibit that enzyme. Okay, so this is a whole class of drugs that have this sulfonamide group in it here. There are other drugs, or there is another drug called trimethoprim that's not a sulfa drug, but it happens to inhibit the second enzyme that's necessary in that pathway. So typically, we don't just administer a sulfa drug. We often administer a sulfa drug in also along with trimethoprim. Okay, so that combination, sulfamethoxazole and trimethoprim, we abbreviate that SXT. So that's one of the antibiotics that you may have used in lab, if you remember, a few weeks ago. Okay, so it creates a kind of a synergistic effect, kind of a 1-2 punch. You take out the first enzyme, and just in case that didn't work, you take out the second one. All right, the trade name, it's confusing. A lot of drugs have, you know, their name, and then the company that manufactures them puts another name on it. So the trade name there is Bactrim. You may have heard of that, not bacitracin, but Bactrim. That's, again, just this SXT sulfa drug. All right. So just to look a little bit more closely at that mechanism, here's your book's graphic here. And at the top, you have several different sulfa drugs, and you can see they have a slightly different structure here on this end, but they all have this ring structure here that's exactly the same as para-aminobenzoic acid, which is the substrate in that folic acid synthesis pathway. Okay, so the reason that's important is because this is how this diagram on the left here, this is how folic acid is supposed to be synthesized. So PABA binds here to the active sites of the enzyme, and then eventually what you end up with is folic acid, right? There's a few more steps in that process. And again, here's the active site of the enzyme, where that substrate binds. Okay, but if there is lots of sulfa drug in that medium, then PABA can't bind into the active sites, and you are not going to get folic acid. So we'll just call it a sulfa drug, okay, that binds there instead. All right, so what kind of inhibition do you call this? Competitive inhibition, right? Where inhibitor binds to the active site instead of the substrate. Okay, so then the product doesn't get formed. All right, the other kind of inhibition is allosteric, but that's if it bound to like another site on the enzyme. Okay, so you know, here's just a kind of a simplification of that pathway. End result here is you get DNA and RNA. So again, without that folic acid, then the cell can't grow, cell can't reproduce. All right, and remember, we get folic acid from our food. That's why we don't have this pathway. We just get it in things that we eat. All right, so just a little bit more about the drug. This is totally synthetic. We discovered this in the 1930s. Gerard Domach, that was a name that we mentioned earlier when we were kind of talking history. Really a miracle drug at the time. It was only effective on streptococcus infections. So limited use, but worked really well against things like strep throat, scarlet fever. At the time, he got the Nobel Prize for it. Now sulfa drugs are a little bit more broad spectrum. They're not used a whole lot because they actually have a little higher, we have a higher incidence of allergies to them, even more so than beta-lactam drugs like penicillin. Somewhere between like 3 and 10% of us, it's estimated, are allergic to sulfa drugs. So they're generally not the first thing that's prescribed. But for some infections, they are a little bit more commonly prescribed, sometimes urinary tract infections or acne. Certain protozoan infections apparently they respond well to. Still using vet medicine quite a bit, but okay. And again, usually allergies, usually a little rash shows up as kind of the first sign of allergies there. All right, so I threw a lot of different drugs at you, and I earlier showed you this concept map that was kind of partially filled out. So at some point you can go in and kind of fill in the rest of it, the drugs that I want you to be, that I expect you to be responsible for. There's a lot of other ones in the book that they kind of touch on. It helped me to just go through and write out this concept map several times to try to keep all those mechanisms of action straight. I think that might be a little bit more helpful than flashcards if that's something that you've used in the past, but just kind of practice writing that out, and I think that will be helpful. Our discussion of antibiotics wouldn't be complete though unless we talked a little bit about antibiotic resistance. All right, so before the pandemic, this was really, most infectious disease experts would say this is the greatest, crisis that we have in treating infectious disease by far, or even biggest health crisis, is the emergence of antibiotic resistant strains of bacteria. Because as we start to run out of antibiotics that are useful, that doesn't just impact being able to cure ear infections and strep throat. That really makes a difference in surgeries that we can do. There's not a ton of surgeries that people have, like knee replacements or hip replacements, that would not happen if we didn't have a way to control the possible bacterial infections that could result from that. So it would definitely, our quality of life would definitely get worse if we didn't have, antibiotics to rely on to get us through things like that. So I want to talk a little bit about how we think bacterial resistance kind of develops in a general way, what we can do about it, and then we'll look at kind of the specific mechanisms, like what exactly is that bacteria doing to keep it protected from the antibiotic. So a few different mechanisms that we'll talk about there. All right, so in general, we think that, say you have a population of bacteria in a petri dish. And of course, bacteria, they reproduce really quickly. Their generation time is pretty short. So, they're copying their DNA all the time, and their mutation rate is going to be higher than, say, what it is for us, right? So, we get a lot of mutations, and every once in a while you might get a mutation that's actually beneficial to the organism. You might get a drug-resistant mutant. So, but oftentimes we think those drug-resistant mutants maybe aren't quite as fit. They get out-competed by other organisms. So a lot of times those probably just, they don't persist. They don't become like the dominant strain. But if there is selective pressure, so if those organisms that are drug resistant are in the presence of the drug, so say there's some selective pressure, meaning exposure to an antibiotic, okay, then those drug-resistant mutants are going to be the only ones that survive, and all the rest of the bacteria will die. And so they can sort of become you know, the dominant cells in a population, and they can persist over time. So examples of resistant organisms, it gets to be kind of an alphabet soup, the way we name them. But this one you probably heard of because we talked about it already, methicillin-resistant Staphylococcus aureus, or MRSA. Not as prevalent for some reason, we're not really sure, than what it was maybe 10 or 15 years ago. But that one's out there. Vancomycin resistant enterococci, VRE, another one that's a real medical danger. We think somewhere around the number that I have, about 20,000 people a year die of antibiotic resistant infections. So people that wouldn't have died if we would have had an antibiotic that could take care of whatever infection that they have. So it's a real problem. There's not very many organisms or not very many bacteria where we say, oh, we don't have problems with resistance to this. Especially tuberculosis, that's another big one where we have strains, about 1% of all the new strains of tuberculosis, or all the new cases of tuberculosis are multi-drug resistant, or we don't have any drug that works on it at all. So it's definitely a huge problem. What do we think some of the things, how did we get here? Alexander Fleming predicted that this would happen even, you know, back in the 40s, that resistance would be a problem. What has contributed to where we are now? Well, just natural selection. So especially in healthcare settings, we're using a lot of antibiotics. That selective pressure is there. I think education is better about this now than what it was 10, 15 years ago, but they have been misused in healthcare. You know, there's pressure on doctors to just, you know, do something to make the patient better. And a lot of patients will put pressure on their doctors and say, you have to give me something. My kid is miserable. You got to give him something that will make him better. So again, I think people are a little better educated about that now. Certainly we have overused them in agriculture, and you can hardly blame farmers. Their profit margin is really small, and anything they can do to increase their profits a little bit. If everybody else is doing it, you almost have to do it as well. So it almost has to be legislated that shouldn't be done. California, just a few years ago, legislated that farmers can't use antibiotics prophylactically. So, you know, hopefully that will spread, but I don't know if any other states have done that yet. And worldwide, in other countries, you can get antibiotics just by walking into a drugstore and buying them off the shelf. So people take them the wrong dose, and they don't take them as long as they're supposed to. They might just take them for a couple days, and then it's not enough. It only kills, you know, some of the bacteria and doesn't entirely get rid of the infection. So that's a problem as well. What can we do about it? Well, that's a great question. There's a few things that we can do. When we are prescribed antibiotics, you should take the full prescription, and encourage people you know to do that as well. Usually after a couple days, you're starting to feel better, and maybe if there's some side effects like diarrhea, people might stop taking them, but the infection's not totally gone. And it could come back even worse, and you know, may give rise to some antibiotic resistant organisms. My dad is absolutely the worst at this. He's prone to gum infections. He'll get a prescription, and he'll take 8 days of it, and then he'll save a couple days for the next time he gets an infection. That's exactly what you're not supposed to do. So make sure you get it totally gone, take your full prescription, unless the patient has some kind of reaction, obviously, then contact your doctor. And this is also a tough one. We want to limit the use whenever possible. So only take them when you need them. I'm not saying if you have strep throat, tough it out and don't get an antibiotic. I'm not saying that, but you don't really, ideally, you don't want to be on antibiotics unless you're sure that you have a bacterial infection. You know, you don't obviously take antibiotics if you have a cold, you know, just in case. Okay, so we want to only take it when we need it. We have found that taking two drugs together is often helpful to make sure we totally get rid of it. So, for example, we call this synergism. So when one drug enhances the effect of the other, you can see here's two antibiotic discs, and you can see the zone here where they are interacting together, even more bacteria are inhibited. So sometimes one drug will make another drug work better. Not always. If you're using a drug that stops growth with penicillin, okay, penicillin needs actively growing cells to work. So that wouldn't necessarily be the case. You can have one drug be antagonistic to another. But sometimes you put two together and they work better. Some examples of that, we talked about adding clavulanic acid to amoxicillin. Clavulanic acid just is a beta-lactamase blocker, and we think that has extended the life of amoxicillin. We talked about trimethoprim and sulfamethoxazole, using those together. Sometimes we use combination of drugs, you know, just again to try to decrease the chances that resistance will develop. So we always give, you know, combinations of drugs to treat TB, especially. You know, we're going to keep doing research to try to find drugs. A few years ago, a speaker came to Grand Valley and was talking about her research with tuberculosis, and she had found a protein that only Mycobacterium tuberculosis made. So she was trying to design a drug to that particular protein. So if we can design a drug that to only work on one, you know, specific organism, then we think that that might be able to, again, lessen the chances that it would become resistant. All right, so how do bacteria actually acquire that resistance? So sometimes it's a random mutation, but we think more often than not, they acquire the information to become resistant from another organism by horizontal gene transfer. So remember our three different kinds of horizontal gene transfer. We have transformation, where a cell that is dead releases its DNA, and then another cell takes up that DNA. We could have transduction, where a piece of DNA is carried by a bacteriophage and infects another cell. Or we think probably the most common way is by conjugation. So by conjugation, most commonly. So remember what conjugation is. We would have a plasmid donor that's F+, that has that fertility plasmid that enables the organism to extend a pilus to another cell, and then a pore opens up between them. It copies its plasmid, and maybe other plasmids that would have that resistant gene, that information, and we'll talk about what that information is in a minute, that would allow now this cell to be able to live in the presence of that antibiotic. Okay, so that's a very effective method to, again, spread that to a whole population of cells. Okay, so we have, depending how you count them, 3, 4 mechanisms. The book actually counts it out a little bit different, but several mechanisms that bacteria can become resistant. One of my colleagues, he used to say, you've heard the term resistance is futile. Okay, so like, you know, aliens come down to Earth and they say, oh, resistance is futile. He used to say, teaching about resistance is futile because students just tended to get mixed up a little bit and it wouldn't sink in. This is fairly straightforward if you just kind of remember what's what. All right, so we have the bacteria that want to survive in the presence of antibiotics. And oftentimes the bacteria will produce something so that the bacteria become not effective. Sorry, the antibiotic becomes effective. Okay, so 3 different mechanisms. The first one is pretty straightforward. If the bacteria can limit the access of the antibiotic into its cell. All right, so how could they do that? One way is to alter the porin structure. So remember, gram-negatives have this outer membrane with lipopolysaccharide in it, as well as these, this is kind of small here, but they have these membrane channels called porins. Okay, so these are, again, transport, membrane transport channels. Okay, and if there's maybe just a little point mutation perhaps in the structure of that porin, those transport channels can become more selective as to what kind of things they allow to enter the cell. Okay, so the idea is the less drug that you let inside the cell, the less likely or the more likely that the cell's going to survive. Okay, so by kind of messing with that porin structure a little bit, less of the drug can actually enter. Along the same lines, sometimes the cell, the antibiotic will be allowed to enter the bacterial cell, but then it will acquire the ability to pump it right back out again, right? So we call that an efflux pump, okay? And a lot of cells already have the ability to do this when they're in the presence of some kind of toxin, for example. The toxin will come into the cell, but then they have the ability to pump it right back out again. Okay, so this would be an active transport using ATP. So we think that this is the way that some cells can become resistant to lots of different drugs. Again, they just pump it right back out again. So both of those ways are limiting access of the antibiotic to the bacteria cell. Okay, another mechanism, we talked about this mechanism already. If the bacteria can make an enzyme, one example is beta-lactamase, that will alter the drug in some way so that now the drug can't bind. Okay, so remember that beta-lactamase enzyme is one that cleaves that beta-lactam ring and opens it up enough so that now it can't bind to that enzyme that's necessary to make the growing cell wall, those transpeptidases. Okay, so again, this is an enzyme that the bacteria make. I kind of searched through the literature to find another example of some kind of modifying enzyme that the bacteria could make. And I found one where they found, I don't remember what kind of bacteria it was, but they found that the bacteria could just alter the functional groups of a drug. So here's a drug, gentamicin. You can see by the structure here, this is an aminoglycoside. And they found bacteria, they could just alter the functional groups a little bit. Like one example, they phosphorylated some of these hydroxyl groups. Or they altered the amino groups a little bit, added a adenyl group to an amino group. Just a slight modification of the structure of that gentamicin made it so the gentamicin couldn't bind to the small subunit anymore. Okay, so again, enzymatic inactivation of the drug so that it can't bind and harm the bacteria. And the third mechanism is that the bacteria can change itself so then that the antibiotic can't bind anymore. All right, so some examples here, ribosomal RNA methylation. Okay, so they acquire the ability to add methyl groups to some of their ribosomal RNA. Okay, and that's actually, remember, what's in the ribosome that the drugs actually bind to. So if they can change the binding site of where the drug binds, okay, then the drug can't bind anymore. Okay, so in other words, the drug can't bind properly to the ribosome. It doesn't alter the function of the ribosome at all, just by adding a few methyl groups apparently, but it can keep the drug from binding. And a few other examples, they found organisms that have had just a point mutation in the binding site. So for example, a point mutation in the DNA gyrase may not affect the activity of the enzyme, but it may alter the ability of the fluoroquinoline drug to bind to it. Okay, same thing, a point mutation in the RNA polymerase. The RNA polymerase could still function perhaps with certain mutations, but then the frampin can't bind to that enzyme anymore. Okay, so these are all examples of just small changes, like the actual mechanisms how the bacteria survive being in the presence of antibiotics. The fourth mechanism is maybe not a mechanism exactly, but it's recognition that when bacteria are in kind of a persister state where they're not actively growing or they're not growing very much, that they're really difficult to kill. And sometimes we don't understand what happens to cause bacteria to kind of switch into this kind of just surviving state where they're not really metabolizing very much. But we know sometimes when they're forming a biofilm, they act a little bit differently and they just kind of, again, just kind of persisting, not really reproducing very much. They're really difficult to kill when they're like that. And I came upon a really interesting article a few years ago. Some researchers at MIT were trying to figure out how to jumpstart bacteria to get them out of this persister state so they'd be more susceptible to antibiotics. And they found that if like if you're trying to, if a kid is sick and you're trying to get a kid to eat, you don't say, here, have some broccoli. You say, here, won't you eat a popsicle, right? You're more likely to get them to eat. So they did the same thing with bacteria. They gave them a sugar source and a suitable electron acceptor to tempt them into, metabolizing that. And once they were actively, so they basically got them to grow a little bit, and then they were more susceptible to the antibiotics. So there's our mechanisms. This is a little activity, maybe take a look at this, and then I'll show this again next time, but it's just kind of, can you put a name on that mechanism? Okay, so I'll put the right answers in here next time, but what do you call it when the drug here can't get into the cell? What did we call that? So take a look at that, and I'll show that beginning of class next time, but I don't want to spend a bunch of time on that now, okay? But do we have questions? Anything about antibiotics or resistance or something you want me to clarify? Don't be afraid to jump in. Okay. All right. Well, let's move on then. Okay. I think that you'll find this chapter pretty appropriate, considering what we've been through the last couple of years. Pretty applicable, talking about the process of infection. So it's kind of a precursor here as we start thinking about how our body fights infection, our whole immune system. So first we want to talk a little bit and acknowledge that we already have lots of bacteria and other kinds of organisms in our body. We call our microbiome or normal microbiota. So we're going to talk about the role that they play, and then organisms that would harm us. How do they gain access to our body? There's just a lot of terminology basically that we use to describe that. How do microbes attach to host cells? And what are some of the mechanisms they have? What are the strategies that they have to actually cause an infection? A little bit about, we're not going to get too much into host responses other than a little kind of terminology there. We're going to talk about how these microbes leave our bodies in order to infect other bodies. And then just a little bit of epidemiology. We'll talk about reservoirs and how infections are transmitted, to kind of finish up there. All right, so normal microbiota. All right, so we all have indigenous microorganisms that are essential to our health. Let's see. Okay. So, and not just bacteria. The first thing we think of is bacteria, but we also have some viruses. We also have protozoa. We also have fungi. We've all got some yeast in us. And we think that there are some archaea that are considered normal microbiota as well. Okay, so a little disconcerting to think that you might have all those different kind of organisms in you, but we know that a lot of them are essential to our health. Some of them may just be commensals, but a lot of them, like I said, we probably have more of a mutualistic relationship. So a mutualistic relationship is one where both organisms benefit. Where a commensalistic relationship is when one member benefits and one is not affected. I always get my spelling of that word mixed up, so I may or may not spell that right. But so, for instance, lots of mutualistic relationships. We've talked about this a little bit before, but the E. coli in our large intestine, it gets nutrients from us, but in turn, we get some vitamin production from us. We probably get a little bit of extra folic acid that it's made. And a lot of times we don't, this is a hot area of research trying to understand these relationships and how we can get kind of the right bacteria in our gut to help us even more. There's a lot of probiotic drugs out there that say, hey, these are the bacteria that will make you be healthier, but really we have no idea right now what can be helpful and what can't. So don't believe all those labels. The probiotics aren't required to be, I don't think they don't have to have the FDA approval. A commensalistic relationship, when one member benefits and one doesn't, sometimes we think for a while, oh, this relationship is commensalistic, and then we learn more about the relationship and we realize, oh, actually, both members are benefiting. We used to think that the bacteria in our skin, it was just kind of leeching off of us, but now we understand, hey, it's probably out competing other organisms that might be worse. So, okay. Our normal microbiota could be transiently associated with us, maybe, in your upper respiratory tract, you associated for a week or two, and then, it kind of dies out. But a lot of our microbiota is resident, meaning you acquired it soon after birth and you have that species for your whole life. Okay, so it can be transitant or resident, but yeah, either one. Sometimes it's not really clear. This is a nice chart just because it kind of gives you some examples. Nothing to memorize here, but you know, we have totally different kinds of residents in different parts of our body. All right, so for instance, in your lower digestive tract, it's pretty, you know, not much access to oxygen there. So we've got some anaerobes that live in our large intestine, where our upper respiratory tract, you know, organisms that are aerobes there maybe. And of course, we have lots of different things, you know, in our, oh, sorry, upper respiratory tract is what I meant to say there. But, you know, if you look down that list there, a lot of different bacteria and other organisms as well. There are some sites in our body that do not have normal microbiota, as long as we know, as long as we can detect right now. But things like your internal organisms, like, your internal organs, like, you know, your heart, your pancreas, your liver, your central nervous system, right? Your brain, like if there are bacteria there, that's always going to be bad. We think the blood is, but I've read some papers that kind of call that into question, that there may actually be some normal microbiota, or we've detected some DNA there anyway, which is not the same thing as a living cell. But there are sites, again, where we know that we just can't tolerate normal microbiota. Okay, so let's talk a few definitions before we go even any further here. Make sure we're using these words correctly. If we have an infection, that means that you have some kind of microbe as within a host. Okay, so really when you say we have normal microbiota, really we're infected by all these organisms. And of course, they're not doing us any harm. Just because you have an infection doesn't necessarily mean that organism is doing harm. If they are doing harm, then we say they're causing disease. All right, so of course, there's lots and lots of examples of that. So oftentimes disease will follow infection. If you have an organism that causes disease in somebody that's healthy, we call that organism a true pathogen. Okay, if something's a pathogen, it means it has potential to cause disease. But there are some organisms that will only cause disease under certain circumstances. So we call that an opportunistic pathogen. Or sometimes we might just say it's an opportunist. Okay, so we're going to explore that a little bit. Under what kind of circumstances might an organism cause disease when it wouldn't normally? So we're gonna poke at that a little bit here in a minute. These two terms, I don't really like the way your book has defined those. So that's why I just wrote them in there. I didn't make you right. But a lot of times those terms pathogenicity and virulence are sort of used kind of interchangeably. If we were to use them absolutely correctly, whether something has the ability to cause disease, we would call that a measure of its pathogenicity. Okay, so kind of so it's most correct to say, does it have the potential to be pathogenic or not? A lot of times though, we say, well, how pathogenic is it? What we really mean is how virulent is it? So virulence is a measure of the disease causing ability of an organism. Okay, so if we say, oh, this bacteria is highly virulent, that means if you're exposed to it in the right way, you're really likely to get sick. Okay, that organism has a lot of tools at its disposal to make it successful at causing disease. And that implies that it's more likely to cause severe disease. Because if we say something is avirulent, it's not dangerous at all. And we also measure virulence based on case fatality rate. Okay, so if it's likely to kill you, then it's going to be highly virulent. Now, your book doesn't define it that way. It says, oh, just because it's likely to cause disease, it doesn't necessarily mean it's severe disease. But that's the part that I take issue with, because again, we measure it based on case fatality rates, then it sort of implies that something that's really virulent is quite dangerous. Okay, so just to kind of try to tease that out a little bit. And then the virulence factors, we're going to give lots of examples of these. These are traits that say bacteria has that make it successful in causing an infection. So maybe it's a toxin, maybe it's something to inhibit our immune system. Maybe it's something to help it bind to the correct cells. Okay, maybe it's an enzyme that it secretes. So we're going to look at lots of examples of virulence factors here as we move forward. All right, so let me go back to that term, opportunistic pathogen. Okay, so sometimes those maybe bacteria on your skin, maybe there's a certain circumstance where they're not benefiting you. Maybe they will turn against you. So when can that happen? Well, if they're introduced into a site that they don't belong. All right, so you get a cut in your skin. Now suddenly the bacteria on your skin have access to your bloodstream. Okay, now maybe they can start to cause some damage. Say somebody has an appendix that bursts. Now suddenly all those bacteria that were stored in that appendix have been released into your body cavity, and that can be a potentially life-threatening situation. Okay. Maybe the host is immune suppressed. Okay, so what kind of things can cause immune suppression? We'll just say extreme age. Okay, we know that as we get older, our immune system wanes a little bit. Young babies don't have quite as developed an immune system, so they maybe are more potentially going to have problems. We could say genetic defects. So there are some people that have certain aspects of their immunity that they are not good at producing. So something that normally wouldn't make somebody else sick maybe has the potential now to make them sick. If they're on some kind of immunosuppressant drug, I think there's two S's there. Okay, that makes a person like prednisone, for example, that can make somebody a little bit more likely to now become sick. Physical or mental stress. How often do you get sick after finals week? Okay, that happens. Maybe you have some other infection that can kind of weaken you in some way that makes you more susceptible to a second infection. And certainly anybody that's malnourished makes them more susceptible to an infection. And we also know that normal microbiota, we talked about this before, if anybody that's on a broad spectrum antibiotic that kind of cuts down on their variability of their normal microbiota, then they can be more susceptible to, say, the C. diff that's normally in their large intestine to overgrowing, or some maybe fungal infection in their mouth or vaginally. Okay, so that's a big one, especially turning our microbiota that's normally there and not hurting us, it can turn against us. Okay, so what other factors do we need to think about? Well, what species of organisms are we talking about? How many of them are is the host exposed to, and how healthy the host is in general. So I show this picture because I know not all the labs do that, but I had my lab go around and streak surfaces and see what grew up on a plate. And I didn't have anybody that had that much variability. But sometimes people get really grossed out by this and say, oh, look what's on my water bottle, I'm going to throw this away. Okay, well, remember the organism maybe that we got to grow up from your water bottle, we're growing them up in much larger numbers than what were on your water bottle. Okay, so you're not exposed to that many. They're kinds of organisms that you're around all the time. And so there's sorts of organisms you kind of need to worry about. And if you're generally healthy, then that's something, not to worry about too. So, occasionally I pick things up off the floor and eat them. I'm going to be honest about that. So, keeping these things in mind. Okay, so thinking about what species of organism, you're exposed to, it makes a big difference, obviously. As to whether that organism is likely to harm you or not. There are some organisms that now, these at the top, we would consider more virulent. That means if you're exposed to them in the right place, most of the time you're going to get sick. Okay, so things like, we mentioned this infection once before, tularemia, rabbit fever. If you happen to get exposed to that bacteria that's carried by the fleas of rabbits, then you are likely to get sick, and that's a pretty serious infection. Yersinia pestis, a causative agent of bubonic plague, right? That's another one. Really successful organism that's likely to make you really sick. Whooping cough sometimes would be called more of an opportunist. A lot of us can carry whooping cough in small numbers and it doesn't make us sick. Problem is when we're around babies, then they are more likely to get sick. So we can kind of be carriers. Pseudomonas is more of an opportunist as well, only when some of maybe our host defenses are down. Burn victims, for example, who get Pseudomonas infections. Again, C. diff, more of an opportunist. And then, these down here almost never would make you sick. They're just less virulent. The lactobacilli, they're in your yogurt. Okay, that's not something that's going to make you sick. Again, Candida, that's something that probably would only make you sick unless it was in really high numbers. Okay, so the species of the organism makes a difference, and how many of them that you're exposed to. We call this the infectious dose. And sometimes there are some species of organisms, we have no idea what the infectious dose is because we can't give that dose to a lab animal and see if it makes them sick or not, right? There are some organisms that, only humans are their only host, so we have no idea. But sometimes we just have to kind of estimate there are some more infections that we think being exposed to one cell is enough to get you sick. Others, like cholera, takes like a billion organisms that you drink. It takes a lot to make you sick. So it kind of depends. I read a case study once where there was an outbreak of some food poisoning at a restaurant, and they were trying to figure out who ate what and how much of what. The guy that was the most sick had eaten 25 pieces of garlic toast. He was really, really sick. Other people that had only eaten a piece or two weren't as sick. So the infectious dose can matter. Here's an organism that has a really, really small infectious dose. If you are exposed to only a few particles, you are likely to get sick if you're not vaccinated. Any ideas what this might be? Probably have never seen anybody with it, but that's somebody with measles. Okay, one of the most virulent pathogens that are known And that's why we got to keep our vaccination up. All right, so we will pick up there next time and see what we can get through. Have a good day. Enjoy your week.